Welcome. In this session, I'm going to detailedly discuss about linkage and recombination. Then after which I will be discussing about polygenic inheritance. Okay. Uh, let's start this session with linkage and recombination. So already in the last session, I have explained what actually is this recombination and uh, linkage. Okay, so here the linkage was discovered by T.H. Morgan. So he's the one who discovered this linkage. Okay, while working on Drosophila melanogaster. So while working on Drosophila melanogaster. Okay, so here T.H. Morgan started doing experiments just like Mendel dihybrid cross. So how Mendel have performed the dihybrid crosses, T.H. Morgan also started to do dihybrid cross. Okay, in Drosophila melanogaster. So T.H. Morgan conducted dihybrid crosses, okay, on Drosophila, right? And when we complete that dihybrid cross, actually in case of Mendel, so when we know when we have seen Mendel's dihybrid cross, in case of dihybrid cross, so Mendel's dihybrid cross, okay, ratio, phenotypic ratio was 9:3:3:1. Okay, so it's the Mendel's dihybrid cross ratio. So when T.H. Morgan did experiments, okay, same dihybrid crosses, okay, on this Drosophila melanogaster, he did not get the ratio, okay, that is 9:3:3:1. Okay, here usually we get this ratio when two genes are independent. So this ratio is expected. So this ratio is expected when two genes are independent. So the ratio is expected when two genes are independent. So let's take here, for example, the seed shape in case of Mendel's, okay, dihybrid cross that has, okay, round and wrinkled. Okay, so here in this case, right, so this is the chromosome number, okay, 7 I'm going to take. So here this is the location where we find, okay, the gene for this seed shape, that is capital R. Okay, and we know its a pair will be present, okay, at the same location, homologous site. So say suppose here small r. Okay, so this is homologous pair. Clear? Now that's what this ratio, that is 9:3:1 ratio, is expected when two genes are independent. Okay, so if they're present here on the same chromosome and present very close to each other, then you will not get this ratio. So you will get a different ratio. That is what Morgan got. So he did not get that Mendel's dihybrid cross ratio, that is 9:3:1 ratio. So that ratio means the dihybrid cross on the Drosophila. So this ratio is a deviation from that of Mendel's dihybrid cross. Right, so let's see how Morgan conducted experiments on Drosophila. So what he has taken, so he has taken a female Drosophila. Okay, so here Drosophila, they are insects and they are unisexual. I'll just mention, so Drosophila melanogaster. So this is a insect and this is dioecious. Okay, and also exhibits sexual dimorphism. So here means males and females are separate. So what he has taken a female Drosophila, so that has a yellow body. So it has a yellow body and a white eye. And he crossed this with a male Drosophila, and that has a brown body, and red eye. So brown body and red eye. So here this male Drosophila is a wild type, whereas this one is a mutant form. Okay, so this is these characters brown body and red eye are okay wild type alleles. Okay, so this is a wild type. So here what we mean by wild type, the genes that are naturally present in a population, so they are called wild alleles. Okay, genes that are present naturally in a population, so genes that are present naturally in a population, so okay, they are called wild alleles. Okay, and the one that has these wild alleles we call it as wild type. So right, the male Drosophila is having a brown body and red eye. Here one more point, so here the wild type, okay, alleles are represented, okay, represented with the plus symbol, okay, and as superscript. So they are going to mention, okay, the plus symbol, okay, as a superscript on the allele that I will show you shortly, right? So he crossed the yellow body, okay, white eye with a brown body and red eye, and the genes for these characters, okay, so genes for, okay, whatever I have told here for these characters are located on X chromosome. So they are located on X chromosome. Okay, right. So now let's see. So let's see cross A. Okay, so this cross A is between genes Y and W. So I already told you. Okay, so yellow body, so that is Y, okay, and white eye, so this is okay W. So these two genes are located on X chromosome, so they're located on X chromosome. So here I'm going to take the X chromosome, so this is the centromere of X chromosome. Okay, and here is the location of yellow body, and immediately after that, okay, it's present W. It means that yellow and white are present very close to one another. So let's see, okay, the female and male. So here the female will have two X chromosomes. Okay, so this is Y and W. So here also Y, W, right? And the female, I'm sorry, the male will have, okay, so one X chromosome. So here this is a wild type, so hence I'm going to mention with the plus in the superscript. So earlier I have told, okay, so this is W plus, and this is, okay, Y chromosome. So female will have two X chromosomes, so this is X, X, right? And this is X chromosome, and this is Y chromosome. So this Y chromosome I mentioned, okay, in this way, okay, right? And so here during the formation of gametes, we know that this will result in formation of only one gamete, one type of gamete, so YW. And here, okay, it results in formation of two gametes, so here Y plus and W plus, and this is another one, okay, right? And during the formation, of course, these are gametes N, and these are parents 2N. Now this one and this one, okay, and this one with this one. So this part I will show here, that is F1 generation, okay. So this is YW and Y plus W plus, because, okay, these are the two gametes that fused, right? Then coming to this one and this one, okay, so of course this is the female offspring we obtain, and here we'll see, so this is with the, okay, YW, okay, so this is the male, okay. So this is the F1 generation what we have obtained here, okay. So this is the F1 generation. So the gametes, their union have resulted so in this combination. So you can see this chromosome with YW here, and wild type Y plus and W plus. So this is in fact, okay, the wild type, and the other one will be having, okay, yellow and white, so yellow body and white eye, okay. So now we know that we need to intercross the F1 generation, so just like how Mendel have performed. So now intercrossing, okay, so right, the F1 generation. So how I'm going to get, okay, so first we'll see when there is no crossing over, okay. So first crossing over, so here crossing over means, okay, this one comes over here and this one, okay, goes over here. So this is what we call crossing over. So here first I will discuss when there is no crossing over, so later we will see about crossing over. Okay, so when crossing over doesn't take place, it results in production of more parental type. So it results in production of more parental type, means the offspring will be similar to the parents. Okay, if crossing over takes place, then they will not be similar to the parents. Okay, so let's see. Okay, so y w, and this is y plus w plus. Okay, so this is the female and the male. So y w. Okay, right. And let's see this one in a Punnett square. I'm going to put it in the Punnett square. So the gametes formation. Okay, so here y w, and so these are the male gametes. Okay, and coming to the female, so here we have y w, and this is y plus and w plus. Right, so we will be seeing here y w, and this is y w. Okay, so this will be yellow white. It will be yellow white. And coming to this, okay, y w. Okay, and in this case also it is yellow white. Then coming to this one and this one. Okay, so it will be y plus w plus, and this is y w. So this will be the wild type is, okay, wild type is wild type. Now this one, okay, here y plus w plus, okay, one. So this one will be, okay, also the right wild type. Okay, so this is you can see in this case. Okay, in case of cross A, they have obtained parental type. So this parental combinations. So parental combinations means, okay, so these are parental combinations. So when we see, okay, so those things, so this is a parental combination. Okay, and this is also a parental combination. So when they have seen the parental combination, the percentage they obtained was 98.7 percentage. So the parental combination they obtained was 98.7. Okay, so it means that more parental combination is because of no crossing over or less crossing over. Okay, so because of that, the parental combination will be more than recombinants. Okay, so recombinants, that I will again tell you. So to understand the recombinants, we need to see the crossing over taking place, okay, between the parents. So this I have told the parental combination, so where they have obtained 98.7 percentage. Now we'll see with the crossing over. So we'll see with the crossing over. So if crossing over takes place, it results in the production of recombinants. So it results in the production of recombinants. Okay, so here crossing over taking place between these two. Okay, so here it is Y, here it comes, okay, Y plus. So here, right, and of course that will be, okay, so that's it. So here, so this is the crossing over which we have seen. Okay, so this one has come up here and gone here. Okay, so that's with the crossing over. So now we'll see a gamete formation here. So we have a Y plus, here W, and this is Y W plus, and in this case Y W. Okay, so these are the gametes we get. Okay, so now I'm going to put them in the Punnett square. The male gametes and female gametes. Okay, so here the male gametes YW, and this is the, okay, Y chromosome, and this one here Y plus W plus, I'm sorry, W. This is the X chromosome, and this is Y W plus. Okay, so now let's see the combinations here. This one, okay, here we have Y plus W and YW, and this one with this one. Okay, so here we get Y plus W, okay, and the Y chromosome, and in this case, okay, YW plus, and this is YW, and here YW plus. Okay, so right, so this is white, so this is white, and you can see in this case the white and yellow, they got separated. So in earlier case, okay, so they were present together, so that is the reason why we have more parental combinations. So in this case the white and yellow, they got separated, so hence in this case we obtained the recombination. Okay, so we have obtained a recombination of 1.3, so that is recombinant type is 1.3%. Okay, so in earlier case we have 98.7%, and in this case only 1.3 percentage we obtain. Okay, it means that, okay, the linkage between yellow and white is strong. So this indicates that the linkage between yellow and white is strong. So that's I'll mention. So yellow and white are tightly linked. So they are tightly linked. So with this experiment we came to know that so yellow and white are tightly linked. Now let's see another cross. So that's cross B. So this is another cross. In this cross, we will be going to see between, okay, genes. So this is between genes, okay, W, that is a white eye, and miniature wing. So white eye, that is W, and miniature wing. So that's M, miniature wing. So these two genes are also present on X chromosome, but the distance between these two genes is, okay, long. So this is the X chromosome, and we know somewhere here is the gene for white eye. So this is W, and here is the gene for miniature wing. So means the long wing is the wild type one. Okay, so here the long wing is the wild type, the long wing, that's the normal one. Okay, so this is a mutant form, that is miniature wing, which means it's a small, okay, and the wing does not extend beyond its abdomen. Okay, so that's the miniature wing, and the distance between the white eye and miniature wing gene, okay, you can see it's very long. In here we have actually is the yellow body. So yellow body and white eye, those genes are very close, so which we have already seen. Now I'm going to discuss about this white eye and the miniature wing gene. So how we are going to get parental combination and non-parental combination. So they are recombinants. Okay, let's see. So here the female, so that has, okay, uh, M, I'm sorry, MW. So in this case MW, and the male, okay, here MW. So here I'm going to take in case of male the wild type. So this is plus plus, this is the wild type, okay, one, and this will be the mutant form, which is white eyed and miniature wing. So white eyed and miniature wing. So let's see, okay, the gamete formation here. Of course, here it results in formation of one type of gamete only. So MW, and here two types of gametes, M plus W plus. Okay, this is another gamete. Okay, so now this and this one, okay, will result in MW. Okay, so this is one offspring, this and this. So MW and M plus W plus. Okay, so we can see in this case we obtained here, okay, white miniature. So this is white miniature, and this will be, okay, wild type, will be wild type. So now I'm going to put those offspring here because we need to intercross the F1 generation. So this is F1 generation, and these are gametes, okay, N, and parents, okay, 2N. So I'll put these two things aside. Okay, so we'll see here. Okay, the female, which is wild type. Okay, so M and W, M plus and W plus. Okay, crossing with the male, so we have M W. So this is a white miniature. Okay, so that's the crossing of intercrossing of F1 generation. So we'll see. So in the first case, no crossing over. So I'm going to do, okay, no crossing over. So when there is no crossing over, we know that the parental combination will be more. Okay, so it results in production of more parental combinations. More parental combination means they more are similar to parents. Okay, so here I'll put this into the Punnett square. Okay, male gametes and female gametes. So here I'm going to take the male, okay, the male gametes first. Okay, so here M W, okay, and Y chromosome, and the female gametes M W and M plus W plus. Okay, so here M W M W, okay, and this one M W, okay, so that's a male, and here this one and this one will have M plus W plus, okay, M W, and this one will be M plus W plus. Okay, so this is what we get. Okay, and in this case, so this will be the wild type. Okay, and here this will be the wild type, and this will be the white miniature winged. Okay, and here also it is white miniature winged. Right, so now the parental combinations. So the parental combination we get is 62.8%. So in this case, okay, we obtained 62.8 type, okay, 8 parental combination or parental type. Now let's see if crossing over takes place. So we'll see, okay, crossing over taking place, and okay, so crossing over, so this one results in production of non-parental combination. So they are called recombinants. So non-parental combination, so recombinants. So now let's put this in the Punnett square. Male gametes, okay, female gametes. So I'm going to take right, so M W, and this is the, okay, Y chromosome, And here, as I mentioned, crossing over has taken place, so this is a crossing over. Okay, so we get m plus w, and okay, in another case we get m w plus. So these are the gametes. So we'll see. So this one m plus w, and this is m w, and here m plus w, and okay, now this one and this one, so m w plus, and here m w, and this is m w plus, right? So this is white, okay, and this is white. So okay, in this case it is miniature, and in this case it is miniature. So here in this case we obtained the recombinant types. So recombinants, okay, so which are non-parental combination. So that's around 37.2%. So obtained 37.2% of non-parental combination, and this indicates, here we can see there are more number of recombinants. So here I'll just put the results of cross A and cross B. So we'll see the results of cross A and cross B. So in cross A, let's see the parental combination and recombinants. So parental combination and recombinants. In that case, it is 98.7%, and recombinants 1.3%. So here in case of cross B, the parental combination. So in case of parental, okay, combination, it's a 62.8%, and recombinants are 37.2%. So you can see in the cross B, which is between, okay, the white eye and miniature wing, as the distance between the genes is, okay, long, okay, they have easily undergone recombination, I mean crossing over, which resulted in recombination. So that is the reason the recombinants were, okay, more in cross B. This indicates that the white eye and miniature wing are loosely linked, okay. So the result indicates, so with this recombinant percentage, okay, the white eye and miniature wing are loosely linked, whereas, okay, yellow body and white eye, so these two are tightly linked. So tightly linked. So this is the, okay, result what we have obtained, okay, after this crossing. So the genes, what we understand here finally, the genes that are present on, okay, a chromosome, and of course they should be present very close to okay, each other on a chromosome, they escape crossing over and they tend to inherit together. and in that case there will be more production of parental combination, and if they undergo crossing over, then it results in okay more recombinants. So we'll say that more linkage, okay, than less recombination. Okay, so less linkage, then there will be more recombination. So this is all about Th Morgan's experiments on Drosophila. Okay, so where he told about linkage and recombination. Now Th Morgan's student, whose name is Alfred Stottiment. So this person, what he did is he used a frequency of recombination. So he used a frequency of recombination. He used a frequency of recombination on the same chromosome, right? And by using this, he came to know the distances between the genes on chromosomes, and thereby he came to know where that gene is present on the chromosome. So that is what so he invented gene mapping. So gene mapping means finding out the locations of, so finding out locations of genes on chromosomes. So how he was able to do this, finding out the locations of genes on chromosomes, is by using the frequency of recombination. Okay, of course that part we will see it in objective. So what is the formula of that frequency of recombination, and how we are going to calculate the distance between two genes and then mapping the genes on the chromosomes. Okay, and so that's the mapping, which means finding out the locations or positions of genes on chromosomes. Right, so I'll just write it. So by using RF, of course, in short, this I call it RF, frequency of recombination. So by using RF, Alfred Stottwand, so he found the distance. He found the distances between genes, and also found the locations, or I'll say the positions of genes on chromosomes. Right, so I told you how it is, all that we'll see in the objective part. Okay, so with this I'll end today's session.